Okay, hi everyone. Thanks so much for joining us today. My name is Tessa Field, and I'm a manager of clinical outreach here at Edimer Pharmaceuticals. I just wanted to go over a few housekeeping things before we get started, and I pass the mic over to Ken. So, as I mentioned earlier, if you're hearing me, hopefully you're all set with your audio. We do welcome your questions throughout the presentation, or at the end we'll have an opportunity for anyone to speak up and voice your questions at that point. But if you have questions along the way, you're more than welcome to use the chat function in the bottom right-hand corner of your screen. Just to make sure that you're all aware, everyone is going to be on mute throughout the presentation, and then when we get to the question portion, we'll go ahead and unmute the line. If you don't have any questions to ask at that point, you can click on the microphone next to your name that will have a line through it and mute yourself again. Otherwise, you can stay unmuted, and you're welcome to ask your questions at that time. Also, please be aware this is being recorded, and we will make a recording available online following this presentation for anybody who may have not had the opportunity to join us today. So we have a diverse audience on the line. It looks like we have a number of folks from different patient foundations around the world, as well as some family members and perhaps even some physicians out there. So we hope that you all find this useful, and we welcome your questions at any time. So without further ado, I'll pass this over to Ken Huttner, our Vice President of Clinical Development here at Edimer, to go ahead and take it away.
Okay, thank you, Tessa. Looking forward to sharing some information with you and talking about XLHED. So let's get started. And the first, here's the team that's working at Edimer. We're based in Cambridge, Massachusetts in the United States, and this is the group that's making this all happen. Okay, so the objective today is to help you understand how we've moved from the first identification of XLHED as the most common of the ectodermal dysplasias all the way to a landmark clinical development plan and clinical studies where we're actually trying to make a difference in the lives of newborn patients with XLHED. So it's a journey that goes over quite a long period of time, and I'll share that with you over the course of the next 45 minutes. So XLHED comes from the group of disorders that are called ectodermal dysplasias. There are at least 200 different types, and they are characterized by involvement of what are called ectodermal tissues, including hair, teeth, nails, and secretory glands, which can be present in the eye, in the respiratory tract, in the digestive tract. So some combination of these gets it to be an ectodermal dysplasia. But if you go to an ectodermal dysplasia book or clinic, most of the patients who are described will have a form of ectodermal dysplasia where they don't sweat normally. Some don't sweat at all, and some just have reduced sweating. And based on these, there's been a genetic analysis, and there are a number of different genes that are involved. But the most common form of the hypohidrotic ectodermal dysplasia, I would say, from our data, 75% of boys who have hypohidrotic ectodermal dysplasia will have the X-linked form, or X-linked hypohidrotic ectodermal dysplasia, XLHED. And the gene has been identified. The inheritance is X-linked, where males are fairly uniformly and severely affected, and females, who may be described as carriers, have a wide range of the severity of their phenotype. So just to summarize what many of you might already know about XLHED, the descriptions go back well over, and partly because it's relatively easy to see, individuals who have thin or sparse hair are missing most of their teeth, and the ones they have are misshapen, and they don't tolerate hot weather because they don't sweat very much. That's the classic triad, but many other medical complications and health issues that are associated. And it's been reported that there's an increased risk of respiratory tract infections, and the infections are more severe with more hospitalizations and antibiotic treatments, with the potential of being intolerant to heat. Often when these patients are undiagnosed, they're at risk of having high fevers, up to 108 degrees with associated febrile seizures. And of course, they're going to be facing many, many years of issues with their dentition. The exact number of patients affected with this disorder may vary from country to country. There's one study that's been published from Denmark, which describes somewhere between 4 and 10 per 100,000 babies born will be affected by XLHED. And over the last 20 years, we now know that the cause of XLHED is a missing molecule called ectodysplasin A1, which is encoded by the ectodysplasin gene and triggers what should be normal development of all these ectodermal features, the hair, the teeth, and the skin features. All right. So once the gene was identified and this molecule was identified as the cause, the idea was a fairly simple one, and often great ideas can be like this, that on the left you see the normal process, and there's a molecule in the right-hand corner of that box that's with the squiggly lines, that is the EDA-A1, and normally it lands on its receptor or sticks to specific cells, and with that initiates the development of sweat glands, teeth, and hair. XLHED, on the other hand, is a condition where EDA-A1 is not present, so there's no triggering of the development of all these appendages. And finally, the solution should be to construct a replacement molecule, not a simple process, but one that can reproduce the activity of EDA-A1, land on the right cells, and if given at the right time and the right dose, and it can get to the right place, then all of these what are called appendages, these sweat glands and the tooth buds and the hair follicles, can all be restored. So this is the idea 20 years ago. And I can share with you now sort of the progress and the journey that we've had coming during that period to where we are today. Now, in the beginning, the thoughts came from several key investigators who were fortunate enough to be working with animal models of XLHED. And one can see them here, that there are animals described, mice, dogs, and bulls who have XLHED, and in some cases the features are very similar to what you see in the patients with XLHED. For example, Axel on the right, a German Shepherd who's missing his hair from his chest, and the hair, a very dry muzzle, and you can see his teeth. He has very few, and they are pointed in all the wrong directions. So having these models is a great advantage for developing approach to treating patients. And the first studies that were published, this going back now to a decade, were based on the idea that if you could replace the missing EDA-A1, and we have a replacement molecule that was developed by Olivier Gade and Pascal Schneider, that you could give this molecule to animals that are EDA-A1 deficient, and if you gave it at the right time, you might be able to correct all the effects that are seen. So this is called the tabby mouse model, and the original experiments were done with the idea that most of development in mammals occurs before you're born. So it was delivered to pregnant tabby moms, and the fetuses then were allowed to deliver and grow. And what you can see on the left is a now adult tabby mouse. It's small, very light coat color. It has a hairless bent one of its molars. On the right, you see such a striking difference that having been exposed to EDI 200 during development, now you have a mouse. They've got the hairs have been restored, the tail is markedly corrected, as are the teeth. So this worked best in the tabby mouse, but also had benefit treating after the babies were born, treating in the first day. This is really a remarkable development. It validates the hypothesis that EDA-A1 replacement can change the course of development in animals with XLHED, but it's quite a step from mice to humans. And so it was important to have a step in between. And what we see here are from a dog model of XLHED, and it's the same basic principle that EDI 200 as EDA-A1 replacement given to newborn pups with XLHED can have dramatic effects. And two of these effects are seen in this publications from Maggie Casal down at the University of Pennsylvania. And you can see in the top the permanent teeth, so these are the adult teeth. You see the unaffected, the 42 teeth that look terrific. And then the XLHED dog has about half the number, and you can see how abnormally positioned they are. Finally, with XLHED treated, meaning EDI 200 in the newborn period, you can see restoration, not completely normal, the dose that was given, but certainly totally restoring the function. When we talk to families and patients where they have an XLHED affected son or brother, one of the other issues is the fact that they make very little saliva, which has a huge impact on their feeding. And the bottom panels show the esophageal glands. These are secretory glands which help make your saliva and help make you help in the digestion. And in the first panel, you see the normal glands of the unaffected dog. And the second panel, you see all the missing glands that are associated with XLHED. And sure enough, if you treat the dogs in the newborn period, and this now is a 1.5-year-old dog, you can see that there's a permanent reappearance of the esophageal glands, restoring all the function in terms of eating and lubrication of the digestive tract. So this was tremendous confirmation and striking that in the dog that you could restore most of the clinical features of XLHED to normal permanently with a short course of EDA-A1 replacement given in the newborn period. So are we ready to go in the clinic with this? No. Before moving forward, it was important for us at Edimer Pharmaceuticals to know more about the natural history of XLHED. So we embarked in a number of natural history studies, and these studies can be surveys, they can be web-based, they can be in person, and they also are opportunities to meet individuals with this with XLHED and to assess them using technologies which might not have been around 10 or 20 years ago, but can give sort of a quantitative assessment of the impact of XLHED on their health. So let me walk you through several of these. We've done 12 of these over 3 years, so I'm not going to talk about all of them. But here's one example. As in collaboration with the National Foundation for Ectodermal Dysplasias in the United States, we participated in setting up a patient-reported registry, all online. This is the largest collection of data on individuals with all the different ectodermal dysplasias, and here I've just picked out males with either XLHED or non-HED. And the striking features here are the ones that were reported 25 years ago by Angus Clark in really the key description of the phenotype, and that shows that the same issue today. There can be associated mortality. Of course, they're going to have issues with dentition, needing possibly implants, that they're going to have issues with sweat, issues with hair loss, increased incidence of asthma, eczema, and other features. When we actually had a chance to get smaller groups and to ask them very specific, we also uncovered some of the XLHED features, which aren't really sure, but families that deal with XLHED know very well. So in this particular natural history study, we focused on respiratory issues. And you can see in the pink bar common issues of hoarseness and asthma. But I would just focus a little bit on something that the literature hadn't really keyed in on, was the common occurrence of early onset nosebleeds that were quite common as a health issue. So the idea that we could actually learn more about natural history studies really was validated with a result like this. As I mentioned, in addition to learning more about the disorder, we were in the process of thinking about treating babies, these would be newborns with XLHED, and focusing on what were the kinds of assessments, what would be the technologies that we would want to use to be minimally invasive but collect the key information. And certainly at the top of our list was assessing sweat. And so here just showing you the two non-invasive measurements that we've validated to use in newborns. On the left is sweat collection, and on the right is a sweat duct imaging device that's the Viviscope 1500. So the sweat collection is a 30-minute collection after stimulating the skin to see if there are functional sweat glands. And the imaging is an opportunity to look underneath the surface of the skin for the presence of normal sweat ducts. And I'll just share with you some of the images that came from this Viviscope. And so these are two images from unaffected male controls, a 5-year-old and a 55-year-old, and it's taken below the surface of the skin, totally non-invasive. It's approved for all ages, from newborns all the way through adults. And what you can see very nicely, these are from the palm. You can see the ridges, which are part of the skin, and on the fingertips give you your fingerprints, but you can see the white dots, which are usually nicely spaced at intervals, and these represent the sweat ducts. And you can see when you're little, they're concentrated because you have all you're going to have from when you're born, and as you grow, they get stretched out, and you can see them spaced further in the 55-year-old. So what do we see in boys with XLHED? Well, we did this at the family conference, and you can see here on the left is the image from the hand of an affected XLHED boy, and on the right from his father, and you can see the marked difference, how easy it is to distinguish with this non-invasive assessment, the ridges that are missing all their sweat ducts versus the normal pattern. So this is something that we can use both to confirm diagnosis, but also to track changes that might occur during the course of a clinical study. And to show that this actually can work on the youngest of patients, these are triplets that we've been following, one of whom is an affected male with XLHED, and they were imaged in the newborn period, and here you can see the two sisters, the middle and the right, where they have sweat ducts, and on the left you can see that the absence of sweat ducts is very evident even in the newborns. So with this kind of information that we have from the natural history studies, as well as the animal studies, it really led us to the position of designing a clinical study plan that we would submit for review in the United States by the FDA and by the different agencies across the world. And the first thing that we had to think about is how would we show that EDA-A1 replacement had a real clinical benefit? And what we did in this situation was to go back to all the literature and talk to the families and experts and identified 7 different areas of where XLHED affected the health of the patient. And you can see them listed here, and these are potential changes, potential endpoints for clinical benefit. And when we went back to the 2 animal models, the tabby mouse and Maggie Cassell's dog colony, of which there's only one in the world, we could find that there was, with EDA-A1 replacement at the right dose and the right time, you could see permanent correction in every one of these endpoints. So it was a very convincing argument to be made with the regulatory agencies that the path really needed to lead to treatment in the newborn period, if not earlier, to get the kind of benefit that would make a difference in the lives of these patients. So with that, of course, you're bringing a new drug, EDI-200, into clinical studies in humans. And for essentially all drugs that are being developed, even if the target and the activity is going to be in newborns, there is always a concern about safety. Safety is my number one priority. It's not Edimer's number one priority, and it certainly is the priority of all the ethics and institutional review boards as well as the regulatory agencies. So as part of the clinical development plan, the program at Edimer started with an adult study. It's called a phase 1 because it's only for safety. There was no expectation or anticipation of clinical benefit. And then only if the data showed that this appeared to be safe to give in humans would we then go on and consider dosing newborns. And when we had the first data, then we would figure out, are we dosing at the right time? Are we giving the right dose? And what would be the appropriate endpoints to bring this into general use? So again, adult study is focused on safety. And then the study in newborns is, of course, will look at safety, but also will have an opportunity to look at efficacy or benefit that the babies might see. All right. So we submitted documents, and I can tell you these are 10,000 pages plus that talk about manufacturing of EDI 200, stability, sterility, how it works in animals, what kind of levels you get with doses. But the number one question that was asked by everybody was, how are you going to select the dose that you want to give? And I want to share just one slide to show one of the approaches we used to have a good scientific approach to answer this question. And the data here shows one of the endpoints in the dogs, and this is adult teeth in the XLHED dogs. And what you can see, I'll walk with you from left to right. The blue bar shows the unaffected dogs, and the adult number peaks at 42. It's the same number as you see in the XLHED carrier female dogs, but in the affected XLHED dogs, the number is about half, and we've seen from the picture that, they're abnormal in shape and position. What we had found in initial studies was one could give newborn dogs with XLHED either a very large dose of EDI 200 or multiple small doses and could see benefit from it. The best benefit, and we felt was the lowest risk, was to give several small doses. And we're testing 5 doses of EDI 200, dosing approximately twice a week. And that worked beautifully starting at day 2, and I'll show you a picture of one of those dogs in just a second. But we needed to know what happened if you didn't start quite, 2 days after a newborn is born. What happens how long is the window at which you might achieve some benefit? This is all dog data, and of course it may be different to some length in humans, but what we can see is if we start at day 2, 5, or 8 in the life of these dogs, you can see maximal benefit. Most of these are just data points from 1 or 2 animals. You can see from this endpoint of teeth, you can see full benefit. Even starting in the 2nd week, the same course of 5 doses, you see significant benefit. And this just shows the benefit of the teeth. We also have benefit in terms of respiratory health and eye and growth. So all things seem to be working if you start within the first 2 weeks, the first week being a little stronger, but the benefit. After that, the differences between humans and dogs can be so great that this is useful, but that's something we may want to test someday in clinical studies. And here, as promised, this is one of the subjects. He was treated as a newborn with 5 doses of EDI 200 starting at day 2, the 10 milligrams per kilo. Has a terrific set of teeth. That's Maggie's hand, and you can see that this dog does have missing teeth. And we have found in general in dogs that postnatal, meaning after he's born, treatment is not good at restoring, protecting hair as one of the features. But the teeth and the respiratory tract and the eyes and all of those benefit. Okay. So with that, we had a plan. We've gone again to agencies both in Europe and the United States, and let me show you some of the studies and some of the results that we have to date. The first study, as mentioned before, is the safety study that was proposed for XLHED affected adults. And any study that's going to involve patient population is listed on this website, clinicaltrials.gov, and this is if you go to that site and you search for Edimer, E-D-I-M-E-R, our company, all the studies that we've done or are doing, including this phase 1 adult study. So in summary, this was adults affected by XLHED, both males and females, and the primary endpoint really was safety. They would receive 2 weeks of dosing, so that's 5 doses over 2 weeks, and then completed would be followed for 4 additional weeks. Now, the first subject received his first dose in December of 2012, so that's less than a year ago, and the last dose was given to the last subject in the spring. And with that, we have all the data, which is reviewed by an independent safety monitoring board. So they are not in charge of looking at, did it work, did it make them, did it help sweating? They just look at the safety, and was the safety profile for EDI-200 in these adults what we expected from animal data, and did it give us reasonable assurance that the neonatal study could be done safely? And so here are the results of their review. First of all, I can tell you that after the dosing is completed, all 6 subjects are doing very well, and there were no red flags. And what I mean by red flag is that there were no unexpected safety findings. When we looked at animals, there was nothing that we were particularly concerned about. There were no heart effects in animals. There were no kidney effects. But the first time in humans is the first time in humans, and it was important to look at everything. So a great deal of monitoring and blood tests was done in order to look at all possible indications of anything unexpected, and nothing was found. What we did learn is that in delivering these doses, so there were 6 subjects, 4 males and 2 females, and each one received 5 doses. So 30 IV doses of EDI-200 have been given, and we learned something about how fast or how slow it needs to be given, depending on the size of the patient, and that it was important to be given at room temperature. Now, the structure of the adult study was very similar to what we wanted to do in newborns. We wanted to give a lower dose and a higher dose so that we could see the spectrum of benefit, because we didn't know which dose was going to be demonstrating the greatest benefit. And so 3 adults got the lower dose, 3 adults got the higher dose. Again, no difference in the rate of any adverse effects or anything that could be related to the EDI-200. And finally, and importantly, when we looked at levels of EDI-200 in the blood, the levels matched what we predicted from animal studies, so that it wasn't going to be a total surprise that when we gave what we thought were low doses to humans, that all of a sudden they would have levels in the blood that were far exceeding what might be safe. So with the safety data and the drug levels and the subjects doing well, the Safety Monitoring Board reviewed and gave us the green light to go ahead and submit and to start enrolling newborns. So we submitted for the Phase 2, the newborn clinical study, in the spring of 2013 once we had the safety data. And let me tell you a little bit about this study. Again, you can find the description of it on clinicaltrials.gov. And currently we're proposing to have 6 sites, 3 in the United States and 3 in Europe. This actually, could change, but this is the current proposed plan. And it looks like a very simple study design based on what we've learned from the animals, that we start with term-affected boys, that they have to have a confirmed diagnosis, so they have to have an examination that looks like XLHED, and they have to have genetic confirmation of XLHED. safety at the top of our list, but also looking at blood levels. And in the end, we want to see, does EDI 200, the EDA-A1 replacement, work in patients, in humans, given in the first couple weeks of life. So the first dose, it needs to be delivered by the 14th day of life. And once they start dosing, they'll get 5 doses over 2 weeks. As we saw in dogs, there's a low dose, which gives some benefit in dogs, but not the maximal, and a higher dose, which gives what we see in dogs as the maximal benefit. So standard in drug development is to start at a lower dose, make sure that you feel comfortable with the safety in these youngest of patients, and then in a second group, go with the higher dose. And that's exactly how we designed the study, that we would enroll somewhere between 3 to 5 newborns at the low dose, and then review the data and move on to the high dose, assuming everything looks as good as we expect it will. The safety portion of this study involves 6 months of monitoring, and that's pretty frequent monitoring. So there's a 3 to 4 week period at the beginning where they're in for their testing and their EDI 200 administration, and then they come back every couple months to be evaluated. And after the 6 months, of course, we will follow them every year to look at all the changes, all the benefits, and follow their health as long as the drug is being developed. Of course, when you write out the protocol, it starts to look incredibly complicated. So there won't be a quiz on this, but this just gives you a sense of the timeline, that there's going to have to be some consideration that it might be an affected boy. He needs to have confirmation by genetic testing, and then sometime in the first 2 weeks of life would start with his EDI 200 dosing. The dosing will not be completed by the end of 2 weeks, but needs to be started by the end of 2 weeks. And then this shows the 2, 4, and 6 month follow-ups. On the left are the safety evaluations, which are numerous, and we have in place a great deal of monitoring as well as blood tests. And on the right gives you all the kind of endpoints that we all want to follow so carefully. And again, many of these will be followed over a period of years. Some things we would know quickly, such as restoration of sweating, but the number of teeth we're not going to know in humans for several years. And growth and development, infections will be following over a long period of time. So the current status of this study is that the study is active, meaning we are recruiting worldwide. We have outreach to continents, pretty much every continent except Antarctica, to try to find patients. We're making good progress with that, and we'll be providing regular updates on how that's going, both through our XLHED network and on our Edimer website. And none of this would have been possible without having a number of people helping us. And certainly at the top of the list of our acknowledgments has to be all the patients and their families that have participated in the web-based surveys, have participated in our natural history studies, and are considering participating in the clinical studies. And we've been fortunate to have wonderful patient support groups, the NFED here in the United States, as well as a dozen groups around the world that are just terrific resources, are very positive about what we're doing, and helping us get the message out. Healthcare providers are often those that will be letting us know about families and subjects and referring them to our network, so we certainly couldn't do this clinical development program without them. And we've had terrific advisors, investigators at our sites, as well as expert consultants who help us with the manufacturing and the design of our studies. At the end of the day, studies are expensive, so we've been very fortunate that investors continue to have confidence and believe that this can be a game changer, that this genetic disorder of development can be influenced. We know it can be done in animals. We just need to find the right timing and the exact dose that will work best in humans, and that's what we're all dedicated to doing. So I want to thank all of these groups for being supportive. Now, for families who want to learn more, or anyone on the call who want to learn more, we've spent considerable amount of time in our first few years getting set up to get the word out. And it really started with having the XLHED network. And this is an opportunity, and I would encourage everyone to log in, get your information in there. It's voluntary, of course, but it allows you to find out everything that we're doing, and it allows us to get information out to you as new results come out, results of the studies, new studies that are starting. In addition to the network, we're now building out various resources for families that are pregnant or are considering having additional children. And I think most importantly, there are many families which don't know for sure that they are at risk. Maybe there's certain family members, but there are others that just don't know. And there are families that have newborns that aren't sure if it's an affected boy. So we have put in place a website that you can go to. And with this, it is now free that you can have your genetic testing done on women who are thinking about having kids or are pregnant, and also their newborn sons within the first month of life. And the genetic testing, again, is totally confidential. The information does not come back to us, but is provided to the families, and then they can make decisions about future pregnancies and participation, or it can help with the monitoring of their newborn children if indeed they are affected. So, of course, social media is another way that information gets out, and you can see here multiple ways that you can continue to be in contact with Edimer and all the things that we're hoping to do. All right. So at this point, I'm happy to respond to questions either from the chat box or we can turn it over to people who want to ask them by unmuting.
So for those of you who, this is Tessa again, hello. For those of you who have sent any questions over through the chat, we'll make sure to start with those. Otherwise, we'll go ahead and unmute the lines for everybody on the call, and then you'll be able to ask any questions that you have out to the group. So just kind of, we have a colleague who is going to go ahead and unmute that for us so you guys can ask your questions. So if you are not interested or if you don't have a question at this moment, you can just go ahead and click the microphone next to your name with the X through it, which will help us to avoid any background noise or feedback that we're getting from your computer. Question right now, please just go ahead and put yourself on mute. Speak up, and at the moment it looks like we don't have any additional questions through the chat, so any first questions that come through to the group.
Hi Tessa, hi Ken, do you hear me? I'm Olivia.
I do. How are you?
I'm fine. I'm so glad to hear you today and to have news. Where are you? You have been you have your following and are they in Europe? What are the news?
Yeah, so we've been fortunate that we've had a number of contacts from families that are interested in having kids or are pregnant and might be at risk for having affected boys, and having terrific progress on giving them the information they need and helping them to make decisions in our study. So, we're going to keep, and as we make more and more, then we'll certainly update that and alert everybody about how the XLHED network. But at this point, I don't want to make make any further statements other than it's been great since we've started ago.
Do you mean that as soon as DB second information and
Well, first of all, I respond that we have families both in the in Europe, and again, as we have more information on the progress of the study, some important events that occur, and when those happen, we will certainly let everyone know.
thank you.
Oh, you're quite welcome.
Bien. Finir alors.
Again, I'm happy to answer, any questions about the animal studies, about EDI 200, about, how we're going to be following and what the time course is, how newborns are assessed. All of these things, this is a great time because, the study is now enrolling. Okay, and I think we've got some that are coming up on the chat box.
So it looks like we had a question about whether or not we would include other countries outside of the ones that we listed earlier in terms of the clinical trial. So I'll go ahead and let Ken answer that question.
We've started with clinical sites in countries where we knew we had investigators with an interest in XLHED. And again, we're a small group, although the picture showed that we're growing, and certainly we're open to including other countries where we can get approval from the national regulatory authorities, and we have an interested investigator, and we have a family that wants to have treatment in their country. So it's certainly exclude from any country around the world that has that infrastructure, has that level of interest. And I see another question about the clinical trials we've done on newborns. And again, this is groundbreaking territory to go into newborns. And we have not done in humans. We've done them in newborn mice and newborn dogs, but really going to be the first in newborns.
So while people are thinking further about maybe some questions that they have, I just wanted to put our contact information up on your screens. You have the information there for Ken, Ramsey, and myself. You're more than welcome to reach out to any of us at any time over email. Or we've included the main number to the office here because we do all have some varying schedules. So we figured that would be the best way for you to get a hold of us in a timely manner. So you can give a call in to the front desk here, and then whichever one of us happens to be in the office, we would be more than happy to answer your questions. It looks like we did have one additional question just come through on the chat line in regards to what type of side effects we've seen in some of the animals that have been treated in the early animal studies. So I'll go ahead and let Ken answer that.
Yeah, of course, that's a key question. And both in treating XLHED-affected mice and dogs and in unaffected animals, which one has to test as part of the safety package, there have been no consistent what we call adverse events or that we see associated with EDI 200 given at the doses that we want to give and by the route that. So this has been exactly what and B Farm and all the world have asked us with them, that we feel, as much as we can tell from all the animal studies, that exposed have not had any significant effects on their health, and certainly we of tabby mice and dozens of dogs who've grown to adulthood, permanent correction, and we have not seen any unexpected health issues.
Okay, so if there are no further questions, then we just want to take the time to thank you for joining us today. I know we have folks around the world that are at the middle of their days or at the end of their days, so thanks so much for fitting us in. Just a reminder again, this will be available online within the coming days, and we'll send you a link to that. And then you'll be able to look at this at any time or reference this if you have questions. Also, please do remember to sign up for the if you haven't done already, and please share additional folks that you know in the XLHED community who might be interested in research and the work that we're doing towards the clinical trials for EDI 200. So thank you to